Thank you for coming. My name is Torkel Gren. I'm from Recipharm, and I will talk about metal-coordinated pharmaceuticals, which is a new technology for getting improved pharmacokinetic properties from active pharmaceutical ingredients. Before I get into the details of the technology, I have a few sort of more business slides presenting the two companies that are doing this work together, Recipharm and Synthonics. And I'm from Recipharm, and that's a CDMO, a global CDMO. We have activities in Europe, Asia, and North America. We employ about 3,500 people, manufacture more than 500 products that are sold practically all over the world in more than 100 countries. We also have very broad manufacturing capabilities, in particular for all sorts of dosage forms, from orals to sterile, semi-solids, whatever. And on top of that, a rapidly growing development unit, or development division. And we have a turnover about approximately €500 million. Synthonics, our partner in this work, is a smaller company, US-based, registered in Delaware, led by the past Chief Scientific Officer of New River Farm. They have their science validated by research grants, by patents, and also in vivo studies. However, at this point, and we should point that out, we have no clinical data yet. And the company is focused on licensing, chemistry, and compounds. Now, we think that we're a good team. One part is bigger than the other one, but we think that it's a nice combination with Synthonics with a lot of expertise in complexation chemistry, in drug delivery research, and in developing and patenting new drug delivery innovations. In Recipharm, on the other hand, we're more focused on and have a lot of expertise in development from really early stage, from the molecule into full-scale manufacture. So we have expertise in formulation science, in analytical development in order to do a good formulation work, and also the expertise and knowledge how to manufacture stuff full scale for commercial manufacture, which in my mind is really important because you want stuff that works not only in the lab but also in the big scale. Now, metal coordination, what is that? I think most of you know that a lot of pharmaceutical molecules form complexes with metals, and it's been known by formulators for decades, and it often creates a lot of trouble in formulation. So what is really new here, that we use this phenomenon to get some benefits out of it instead. The requisite for a metal complex to be formed is that you need two heteroatoms in the molecule and such a structure so that you can form a ring with 4 to 7 members, including the metal. So, and if you start to look at pharmaceutical molecules, you'll see that most of them actually will fulfill this requirement. Now, by creating these complexes, you get a new entity which has a very good ability to be patented in an efficient way because it's a really new structure. Also, of course, when you make this complex, you'll get new physicochemical properties and also pharmacokinetic properties. On the other hand, as soon as the metal is split away and the complex dissolves, you get the original molecule and the original biological effect, of course. And by using different metals and different ligands, you can also control the type of properties that you give the molecule. Also, a very interesting aspect of this is, while we have this good possibility to get strong patents, at the same time, from a regulatory standpoint, this is not a new chemical entity because it's not real any covalent bonds involved. So there is good possibilities for an expedited regulatory pathway here. What can you use? What thing can you get out of a metal complex? Lots of different characteristics, but we have evidence that you can get controlled release, You can probably use it for targeted release. We can definitely improve absorption, reduce variability in bioavailability, and we can improve solubility, aqueous solubility, but also solubility in lipids, and actually achieve both at once. You can get amphiphilic properties into a molecule. You can use it to accelerate onset on action, and we'll look at a bit on bioadhesive properties as well. And of course, these complexes can be further modified by applying all sorts of formulation technologies on top of it, like coating or using surfactants or whatever. The first example I will present to you is about levodopa. And levodopa is a molecule that has been known for, I think, at least 50 years. It's used against the symptoms of Parkinson's disease. It was discovered by the Swedish scientist Arvid Carlsson many years ago. And it's old, but it's actually relatively effective in the early stages of Parkinson's disease. You can really get the symptoms down quite significantly. Unfortunately, however, at later stages, the therapeutic windows get very narrow, and it's really difficult to treat the patients efficiently with levodopa. And what you would need here is, of course, a flatter plasma profile, like the like the red line on the picture, instead of what you get with a tablet. Problem, though, is it's not that easy to accomplish extended release with this molecule because it's only absorbed from the upper part of the intestine. So if you make a normal modified release preparation, it will just pass through, and most of it will not be absorbed. What we have done here is to, or really our friends at Synthonix have done here, is to create a complex with bismuth and levodopa. And we think the mechanism here is that the bismuth ions will bind to anionic structures in the mucosal layer. So it will sort of stuck there. And some of you may say, when you see this as I did the first time, bismuth, isn't that really toxic? And as a matter of fact, considering you would think it would be considering its place in the periodic table, but it's a very untoxic metal. So what we get is an ionic interaction of a cationic charged polymeric metal, or the complex, with the anionic charged structure in the mucus. And this will make the drug stick here and then slowly get released and into the bloodstream. At least we think that is the mechanism. You can look at it like this here with the drug getting stuck here in the upper part of the intestine by the bismuth adding to the mucus layer and then getting into the bloodstream. And we have no proof for this mechanism, so it's just a hypothesis that it works this way. However, we have results, in vivo results, and this is from dogs, where we can show that if we compare the black line, which is native levodopa, with the bismuth complex of levodopa, we get a lower plasma peak, and we get a longer, higher plasma level, more close to the ideal level. So we think this is encouraging data, but of course it's still only in dogs. So what we're doing right now is a lot of development work on it. We have had preliminary guiding discussions with the FDA that thinks that yes, this would be eligible for a 505(b)(2) pathway. We are working with the drug development at the Recipharm site in Sweden. We have performed preclinical tox studies. We have made API, and also that has been done in Recipharm's facility in Italy, in Milan. We are now manufacturing GMP material at Recipharm in Sweden, Solna, and we have submitted a IMPD and plan to perform the first in-man study later this fall. So hopefully, not so long away in the future, we will really see if this works in man or not. The next example I have here is metallothiodothionine, and I always stumble when I'm trying to pronounce it, so I'll say T3 in the rest of my presentation here. And also in this case, there would be a need for modified release formulation. And the clinical need is really that you would like to have a slow release, a modified release of T3 to sort of mimic the plasma level in healthy people. Unfortunately, that has shown to be really difficult to achieve. You can get extended release preparations with T4, which is the precursor of T3. And it works well in a lot of patients, but not in all of them. And supposedly that is due to that not all people, or the conversion of T4 to T3 doesn't work that well in everyone. So a certain fraction of the patients doesn't respond that well to this treatment. And this is really early for us, but so far we have at least data in rat and show that we get a flatter plasma profile, the red line in comparison, and this is with a zinc complex of T3 compared to normal sodium T3. So we have actually shown that at least in animal you can flatten out this the plasma level. But of course, still very early data. I also mentioned in the beginning that solubility enhancement would be an opportunity here. And this is just one example where we have looked at furosemide, and you can see that, and we looked at different solvents, and with magnesium furosemide, of course, you get a significant increase of solubility, but you can get even more with the complex, and not only a solubility enhancement in aqueous environment, but also in other solvents. So, but this remains to be explored a lot more, I think. So I would like to conclude and sort of summarize this by saying that for sure metal coordination is applicable to a large number of molecules. And then of course, I'm not saying that with all molecules you can achieve exactly the properties that you would like, but there is a lot of opportunities to modify physicochemical and pharmacokinetic properties. And metal coordination is compatible with other formulation technologies, so you can add additional enhancement of what you achieve with just the complexation in itself. Many different properties can be improved. We've looked at modified release and bioadhesion here. And the combination of good patent protection in combination with a rapid regulatory pathway could be very interesting for a lot of molecules. From a manufacturing standpoint, this shouldn't be that complicated because the equipment you need to manufacture the complex is the normal type of equipment that you use in API synthesis. You need a reactor, and that's it. And also, if we look at the drug product manufacture, the same technologies like tableting, coating, capsuling, whatever, will be applicable also for these complexes. And in many cases, it's relatively easy to at least do an initial in vitro check. Will this concept work, or is there any chance to get work for this molecule, or is it not? So it's a technology with a lot of opportunities, we think. It's still very early, but if you're interested to discuss this more, I would be very happy, of course, to do that with you, for instance, in the Recipharm stand at 3:30. I'm really grateful that you wanted to come here and listen to this, so thanks a lot for your attention. Questions, I would be really happy to try to answer them at least. Yeah.
coordinated organic compounds that have some solubility problems, there is an advantage for control release modification of such a drugs. But if you aim to produce a parenteral formulation, but a control release parenteral formulation, possibly there is a difficulties with the solubility of such a material, this metal coordinated material. What you recommend in this case?
So I guess the question is that if we have a compound that has too low solubility,
Yes, right.
and you want to make an injectable preparation,
Yes.
what to do? So I guess my first advice wouldn't be to go for metal complexation because it's a new technology. So I would try the standard technologies first, of course, because that would be a more rapid and less risky way to a successful product. But of course, if you can't achieve what you're after with cyclodextrins or with co-solvents and so on, then I think this would be interesting. And of course, if we are successful now, in a few years, this technology will be much more attractive because then it will be less risky when it's tested. Okay, thanks a lot.